Hello, everyone. I am so excited to be here today as part of this amazing panel of experts to talk about the connection between oral and systemic health. Today, I'll be discussing the relationship between the oral GI microbiome and the totality of care, incorporating some of the concepts you've already heard or you will hear from our other excellent presenters. Before I begin, let me give a brief conflict of interest disclosure. I am an employee of ProBioRa Health, but this company's products and services have no influence on the content that I am presenting here today. I have some goals for you today. My overarching goal is that you walk away from this presentation and all of the entirety of this symposium feeling empowered with the knowledge about the connection between oral and total body health. But from my brief presentation, we'll narrow that down just a bit. After this, my goal is that you'll be able to define the role of the oral and GI microbiome for total body health, that you'll be able to explain the difference between species and strain as they relate to probiotic applications, and that you'll be able to compare the application of probiotics for both oral and GI health. In order to meet those objectives, I've organized today's talk to take us back in time just a bit to remember our history and how we've arrived at the state of oral and systemic health of today. Then we're going to discuss how bacteria play a role in oral health, including the good, the bad, and the ugly, followed by the overarching theme of my presentation, which is the connection between the oral and gastrointestinal microbiome. We'll close with some microbial concepts for preventative care and questions and considerations for clinicians, scientists, and the general population to consider if you want to use something like a probiotic to improve the state of oral systemic health. So first, let's go back in time and let's start with a little bit of my history. It's possible that if you took a peek at my bio for this symposium, you may have noticed that I am not a practicing or trained clinician. Instead, I'm a scientist, specifically a microbial ecologist, and I actually got my start working with microbial communities in the environment, not in the body. My dissertation research was about as far removed from the oral cavity as possible. I studied the effect of cattle manure and concentrated animal feeding farms on antibiotic resistance in the environment. So there is a little bit of a health connection there with antibiotic resistance, but again, my focus was really on how the environment can affect levels of antibiotic resistance in the clinic. If you take a look at the picture here in this slide right here, what you're actually looking at is a huge part of my doctoral research. So this machine is actually spraying cattle manure onto a crop field. That manure then sits on top of the crop field. It might sink into the soil, or if a huge rain event happens, might run off into the surface water, which can greatly affect the drinking water availability and cleanliness, especially in rural areas, and may even affect the levels of antibiotic resistance and antibiotic resistant infections. If you're wondering, how did you go from cattle manure to the mouth? It's actually pretty simple. The concepts that we use to study microbial communities in these types of environments and other various environments, such as cattle farms, surface waters, and soils, they're pretty similar to the methods that we use to study the microorganisms and their interactions in the oral cavity. One concept that might be familiar to you is the One Health concept. This has been widely embraced by microbiologists, and it's helped to improve our understanding of the fact that health in one compartment, such as environmental health, affects health in all of the others, such as human health and animal health, and they're all interconnected. And so we call that the One Health concept. This was very clear in my work with environmental antibiotic resistance, and we discovered that many of the antibiotic resistance genes and multi-drug resistant bacteria that we found in the environment were similar, if not identical, to some of the antibiotic resistance genes that we were finding in the clinic. This concept should sound quite familiar, as it's a perfect mirror for how we should be evaluating the totality of care. Each body system can have a profound effect upon the others and shouldn't be studied alone or in a vacuum. Just like antibiotic resistance in surface water can appear in clinical infections, oral health issues such as gingivitis can have wide-ranging health effects in other body areas, including immune disease, cardiovascular disease, even things such as cancer. And therefore, the best way to approach a complex concept, such as the relationship between the oral cavity and the totality of care, is really to create this beautiful intersection of knowledge where dentistry, medicine, and science come together to share information, concepts, and challenges that help broaden our understanding of how seemingly separate body systems interact. And because those intersections are so critical, I like to say that this is where the magic happens, is at the intersection of these three. So I'm really excited to be here today to discuss a little of what I know about the science of the oral cavity and how it relates to the totality of care. So enough about my history. Let's go back much, much further in time and start with a very simple observation. I want to know, does anybody recognize this image? I'm hoping it's a yes from some folks here at the symposium today because this is the original dental plaque specimen and the very first bacteria ever observed or drawn by the one and only von Leeuwenhoek using his very sophisticated scopes. Keep in mind, he was a textile distributor, right? But this observation of bacteria, starting with his dental plaque, which we see here, but also in water samples and then a variety of other samples that he took, started an entirely new branch of science. And eventually, it really improved our understanding of parthenogenesis. That led us, about 200 years later, to Koch's postulates, which we would not have been able to have without those initial observations of microorganisms underneath the scope. Koch's postulates were a groundbreaking advancement in disease etiology, right? They provided a method to clearly demonstrate a causal relationship between a microorganism and a disease. However, these postulates were quite limited in scope. Other diseases, such as irritable bowel syndrome, oral ulcers, or gastroenteritis, could not easily be described or linked to a simple one microorganism, one disease state. Additionally, many disease states can't be linked or attributed to just one organism, and they may be driven by complex interactions between microbial communities, both with one another and with their host. Systemic conditions such as heart disease or metabolic disorders can have multiple contributing factors. And so looking at a disease from a one organism, one disease state perspective doesn't really apply to our understanding of the body as it is today. That's led to a new age advancement of Koch's postulates, right? So it's been termed a couple of different things, whether that's Koch's molecular or ecological postulates, but it's really helped us move from a state of a single acting, unsocial organism, right, causing one disease, to a better understanding of the fact that most microorganisms interact strongly and they tend to build up these very stable networks of structures, okay, that interact with their host and the environment in a variety of different ways. One simple example of this, going all the way back to von Leeuwenhoek, is plaque, right? Plaque can build up on the teeth, but plaque can also build up in the heart and plaque can build up in the brain. Plaques are very complex structures that can include microorganisms, but also their byproducts and other extracellular components that may induce a variety of disease states, such as heart disease and cavities. This is why it's critical to evaluate both health and disease in the body from a totality of care perspective. Today, I'm focusing on the oral microbiome and its relationship to the GI microbiome and how they intersect with this totality of care concept. I am going to present studies and evidence that clearly show that the microbiome as a whole affects total body health, and I'm going to explain how we can use this information for more informed oral health outcomes. I'm specifically going to focus on how we can use microbial interventions, such as probiotics, for good and to help support health by shifting the microbiome from a state of dysbiosis to eubiosis. So let's talk bacteria in the mouth, the good, the bad, and the ugly. First, we're going to start with the good. If you want healthy teeth and gums and an overall healthy mouth, you need some essential bacteria. They're critical for multiple functions in the mouth, including the start of breakdown of food, helping prevent infection and inflammation, and they also produce really important antimicrobials or other biocidins that help control pathogens in the mouth. As a young child and even a young adult, my only interaction for many years with dentists was at my annual cleanings, where I often heard that we want to remove plaque and reduce bacteria in the mouth. At the time, what I understood this to mean was that bacteria are bad and something that we should get rid of at all costs. Obviously, I went through college, became a microbiologist, went on to get my doctoral degree in microbiology. So I now better understand this and realize that's probably not what my dentist was trying to tell me. Instead, he was communicating that we're talking about reducing bacteria that cause disease like gingivitis and cavities, and the different methods that we can do to help prevent or eradicate those pathogens from coming back or attaching to our teeth. I also, have since learned that we have a strong population of commensal bacteria, and those include members of these genera that you can see over here on the right, such as Streptococcus, Neisseria, Rothia, that are really critical and important for supporting healthy teeth and gums. These essential microorganisms are who we consider the good guys. These are the bacteria that you want present in your mouth at high levels in high abundance on your teeth, gums, and tongue. But the oral cavity also has a collection of the bad or opportunistic pathogens. These are pretty common in the oral cavity. They are naturally found in a body site, right, such as your mouth. But when conditions change due to stress, diet, potentially the use of an antibiotic or other factors, an overgrowth of these opportunistic pathogens may occur, which can lead to a disease state. A good example of this in the oral cavity is the bacterium Fusobacterium nucleatum, which is a common anaerobic bacterium frequently found in low abundance in a healthy mouth. But when the mouth enters into a disease state where it's in dysbiosis, the bacterium is found in higher numbers, especially in the early stages of gingivitis, as you can see here. Then we have the ugly. These bacteria are generally only found associated with a disease in a specific body area. They may be location bound. A good example of this is Phoramonas gingivalis, right? P. gingivalis, which is an etiological cause of periodontitis. When it's found in high abundance in the oral cavity, it's typically in a diseased mouth. It can be found in a healthy mouth, but it tends to be found in extremely low abundance. In reality, most mouths have a combination of the good, the bad, and the ugly, right? So we have commensals, we have opportunistic pathogens, and we may even have some of these obligate pathogens. But the important thing to remember is that they're strongly affected by interactions between one another, such as competition and resource partitioning. And they also interact with the changing environment of the oral cavity and the host. So understanding how oral microorganisms interact with one another and influence oral systemic health is critical. In the clinical context, this would be using something like a salivary diagnostic, which we've already heard about in a couple of the talks this morning. It's really important to know and understand which microorganisms are present so that then you can figure out how to treat the microorganisms that are there in the disease states that they may be causing. Now that we understand that the oral microbiome is composed of a complex suite of both commensal and opportunistic bacteria, let's dive into my small piece of the symposium, which focuses on the relationship between the oral gastrointestinal microbiome and how the oral microbiome can affect disease states in the gut. When I think of the oral GI microbiome continuum, I always think of this image, right? This is like the stereotypical image, classical. This is what I saw way back whenever I took like general human anatomy, which has been quite a while ago now. If you take a look at this, can anyone clearly see what's missing for the GI tract in this image? It's missing the oral cavity. The oral cavity should always be considered part of the GI tract as the primary entry point of the body, and because bacteria and pathogens that enter through the oral cavity may translocate along the oral GI axis. In a health context, understanding how closely connected these two body areas are is critical to understanding if the mouth is putting the gut at risk. One way that the mouth may put the gut at risk is via bacterial translocation between these two compartments. Movement of food, fluid, and bacteria happens continuously throughout the day. And can you think of the most common method of translocation of bacteria from the mouth to the gut? That's right, it's saliva. You and I swallow anywhere between 0.5 and 1.5 liters of saliva per day, chock full of oral bacteria. Those bacteria from your tongue, teeth, and gums are transported via saliva to other body areas along your GI tract. And it makes up nearly half of your daily fluid intake. So just the natural movement of saliva in your mouth makes up half of your daily fluid intake that you actually swallow. The movement of bacteria between these two body areas is critical to understand because what we traditionally think of as the gut, right? So your stomach, your small intestine, and your large intestine houses the highest abundance of bacteria in the body. But the oral cavity is a close second in terms of numbers. That's a ton of bacteria moving and interacting, searching for resources and attachment sites that either help support health or result in disease. If there's an imbalance in the microbiome of the oral cavity, it can lead to additional imbalances in the gut microbiome as these opportunistic pathogens get swallowed. And so let's use an example and take a look at a common oral microorganism, Candida albicans. This fungus is indigenous to both the oral cavity and the gut. It's very common in both areas, but overgrowth in the mouth can lead to oral thrush. When Candida spreads to other body areas where it's not native, such as the heart, invasive candiditis can occur, leading to serious health complications. Interestingly, control of Candida albicans infections is being studied, and recent research, we're talking 2022 here, suggests that biological methods of control may help reduce and prevent infections. While they don't call it this on this slide, they call it biological control. This is truly an example of probiotic application. So what they're trying to do is control the level of Candida albicans by using other bacteria that can either produce some kind of antimicrobial, maybe they produce a bactericidin, or maybe they're just much better at growing in that specific body area and they can outcompete Candida for resources. That is what this study was evaluating, whether these bacteria, these antagonists, can help reduce the activity and levels of Candida in the mouth. These type of interactions are common. They occur all of the time in the oral cavity and in the gut when commensal bacteria produce metabolites, byproducts, or outcompete pathogen. These methods of controlling infections are long-standing rules of ecology and species interactions. They're interventions and they help provide a more holistic approach to disease management. Controlling oral infections is critical for GI health because, as we heard earlier, leaky gums can lead to a leaky gut. We heard about this concept in earlier talks today, but I want to emphasize a few points. Most oral bacteria present in a healthy mouth cannot survive the high levels of acidity in the stomach, and therefore they don't pose much risk to the GI tract. However, opportunistic pathogens, such as P. gingivalis, are more acid resistant. When the oral cavity is infected with something like P. gingivalis, it may end up being distributed throughout the body via swallowing, like we discussed earlier, or via inflammation, which can provide pathways for systemic circulation. This type of bacterial translocation and interaction highlights how oral disease, such as gingivitis or periodontitis, can have wide-ranging health effects in the body, as shown here from this graphic I showed earlier. This leaky gum, leaky gut effect is supported by recent research. Periodontitis in the oral cavity, periodontitis in the oral cavity leads to an imbalanced microbiome, more commonly called oral dysbiosis, right, leading to an increase in the levels of this pathogenic bacteria. These bacteria are then more prone to translocation, right? And again, whether it's via saliva or inflammatory pathways, they may end up in the gut. When these pathogens end up colonizing the gut, they can attach to the epithelium, right? Similar to the effect in the oral cavity, that's the similar lining they attach to in the oral cavity, leading to more inflammation and what we know as inflammatory bowel disease. So again, this is a good example of how the oral microbiome and oral disease can be directly linked to disease in the gut. Other serious health conditions in the gastrointestinal tract may be influenced by oral bacteria, such as this study that was recently published in Nature. Here, this is like brand new here in early 2024, which showed that a common oral bacterium, again, Fusobacterium nucleatum, one of the opportunistic pathogens we were talking about previously, is highly prevalent in colorectal cancer. When the mice were infected, so you have an uninfected control, and then you have two different clades of Fusobacterium nucleatum or strain types used to infect these mice. A specific strain or clade of that Fusobacterium nucleatum, when the mice were infected with it, resulted in larger numbers of cancer in the large intestine, more tumors. This really highlights two important concepts, which one is that oral bacteria can influence the health and disease status of the gastrointestinal tract, and two, the bacterial strain matters when it comes to health and disease. Here, only this specific strain, this specific clade is what they used in the study of Fusobacterium nucleatum resulted in more tumors, whereas this other strain or clade of the exact same species of bacteria didn't result in the same number of tumors and was non-significant compared to the control. So strain really matters. This concept that strains matter for both health and disease plays a major role in how we might use biological control methods for preventative oral care. Let's bring some concepts full circle. The first is that the oral microbiome and the gastrointestinal microbiome are intricately connected, both literally and figuratively. The second is that when the microbiome of the oral cavity and the gut are out of balance, disease can occur, right? So we might end up in dysbiosis. To treat the underlying disease, we should first test, so coming back to salivary diagnostics, to know which microorganisms are present and the best pathway to treat. Third, microbial dysbiosis in the oral cavity and gut can be controlled using biological methods, right? We saw that previously in the Candida example. And that means that other bacteria or fungi may be able to control the overgrowth of an unwanted pathogen through a concept called and termed probiotics. And fourth, in order to heal our leaky issues, whether that be the gum or in the gut, it's critical to focus specifically on the microbiome and to reverse dysbiosis by using the new concept of repopulating, which would be repopulating with probiotics, which leads to a rebalance or eubiosis, a better health, better state of the microbiome. This is really where probiotics and that concept comes into play. I want to emphasize probiotics, using probiotics for healthcare is not a new concept. You guys are all probably very familiar with that. Probiotics are bacteria that confer a health benefit, and the concept has really been around since the late 1940s. However, today's technology is allowing better characterization of probiotic mechanisms of action, especially with new molecular and sequencing technologies, in ways that just were not available when the concept first started, whenever most of the testing for probiotics was done using biochemical methods. In general, when folks hear the word probiotics, they think gut health. And maybe potentially what their doctor is telling them to take if they're using an antibiotic, right? To help repopulate whenever the antibiotic has wiped the gut, pretty much wiped all of the microbiome out of the gut whenever you're on that broad spectrum antibiotic. Probiotics as a whole work through a very simple concept, right? So you have an irritated, inflamed area. Again, it's generally thought of in the gut, a disease state that's suffering from microbial imbalance, microbial dysbiosis. If you add a probiotic into that system, you can help restore the balance and reach a healthy balance state. Now, the actual mechanism of action of the probiotic can strongly vary. We talked about a few of those before, a few of those microbial interactions that may result in a healthier state, right? So competitive exclusion, production of things like antimicrobials and biocidins. But overall, the concept is truly take a disease state of the microbiome, add a probiotic into the system, which should, if it is a true probiotic, confer some type of a health benefit, resulting in a rebalancing of the microbiome in that area. Probiotic research has steadily increased since the first use of the word in the very first publication here in 1947. I know you can't see the blip because of the scale, but there should be a blip there for one publication in 47. Interest really started to increase though in the late 2000s, heading into the 2010s and 2020s of today because of the omics era. Again, we can get so much more information so much more quickly through sequencing technologies that are now available. Much of this interest revolved around using probiotics in medicine as functional foods to prevent disease, generally in the lower GI or urinary tract, and using them potentially, the concept also has been termed functional foods, in order to prevent disease. And a general interest in the concept of biological control of microbial diseases steadily increased. However, to date, probiotics really haven't gained widespread recognition in medical or dental practice for a very simple reason. No probiotics are currently approved by the FDA for use as drugs in the US. And what that means is that probiotics cannot be labeled as something that can prevent, cure, or treat a specific disease. And they also, but because of that, because they can't be listed as a drug, so that's the negative, right? Even if they can show a health benefit, they have not generally been through enough testing like a drug would have to with multiple clinical trials to be considered a drug. And because of that, on the converse side, they don't have to undergo the same rigor as traditional drugs before introduction into the market. And I will touch on that and why that's a critical point in a few more slides. But overall, probiotics really haven't gained a lot of recognition because they have not been able to be classified and labeled as drugs in the US. While gut probiotics were thought to be a potential medical breakthrough in the early 2000s, oral care probiotics are still a relatively new concept. However, their mechanism of action, again, it's quite simple. So just like the gut imagery before, in the oral cavity, probiotics work based on simple ecological concepts of competition and niche. So if you have a microbial community on your tooth, which we do, and again, I'm just showing the tooth part of the oral cavity, but if you have what we consider to be a nice, healthy consortium, a nice, healthy mixture here of microorganisms in a state of eubiosis in a healthy state, that then has a pathogen introduced, whether that is due to a lifestyle change, right, some external pressure or stressor, or due to an infection that comes from an external or internal source, you lead to that tooth, the oral cavity, entering into a state of dysbiosis or an imbalanced microbiome. But if you have that state of dysbiosis and then you then introduce a probiotic, the concept should work relatively the same. The probiotic is then going to compete with the pathogen for resources, for ecological space, maybe even potentially producing some byproducts that inhibit the growth of the pathogen, reincorporating itself because it should come from the same body area if it's a true probiotic. So it should come from the teeth and gums, be able to attach to those tooth and gum sites and grow well, helping to displace that pathogen, resulting in eubiosis or a more balanced oral microbiome over time. Now, that is a super simple overview. In reality, bacterial competition, inhibition, and production of antimicrobials is much more complicated and involves multiple genes and pathways. However, for a bacterium to be considered a probiotic, it must have one defining characteristic. It needs to provide a health benefit. And again, this all really comes back to what the overarching theme of my talk today, right? The oral GI microbiome and how can we improve the health of the microbiome to help improve total body health. Probiotics are a pretty, not necessarily novel, but an important potential concept to use a biological method of control to return us to that healthy state. This tends to result in a very common question about probiotic intervention. Do probiotics actually work? Keep in mind, like we talked about before, they're not drugs, and because they're not drugs, clinical trial data can be limited. For example, IRBs routinely block studies investigating the effect of probiotics on health conditions such as gingivitis, periodontitis, cavities, but will allow studies investigating the effect of probiotics on the concentration of oral pathogens that cause gingivitis. So it's all about the terminology and the way that you study it, but it really can limit the type of data that can be collected in clinical trials for probiotics. Additionally, this reduces the way that clinical trials are set up, the fact that probiotics are not currently classified as drugs, limits their claims to structure function claims only. So that means that a lot of these products, for example, for oral care probiotics, the bottle will only say that they can whiten teeth, freshen breath, and may reduce or inhibit the growth of oral pathogens. They don't tend to be very specific because they're not allowed to be. So if you are not in a position to really read up on the science behind the specific probiotic strains that are being used in products, you may be unaware of some of the potential health benefits that exist for this natural method of biological control. Despite these factors, the research into probiotic mechanisms of health and biological microbiome control have not stopped. So the research continues. If we go back to the gut, where a lot of this research started, fecal transplants have been shown to increase survival in patients with severe C. diff infections. And this study is actually really fascinating. What it showed, what you can see here on this graph on the left, okay, so what we're looking at here is survival of individuals with severe C. diff infections. The green is if they had a combination of probiotic and medical intervention. The blue is survival if they had medical intervention only. And you can see that by 80 to 100 days, there's a huge difference in the survival when probiotics were included versus medical intervention only. And so this is a really clear example of the fact that a shift in the microbiome can make a huge difference in severe disease. The same holds true in the oral cavity. And so beneficial strains of Streptococcus, such as Streptococcus salivarius, have been shown to reduce the caries risk in children when the strain was introduced into the oral cavity over 90 days. And what you can see, this is really well shown by the pie chart here at the top. When Streptococcus salivarius was introduced at day 0, here was the chance, green was the chance to avoid new cavities. But by day 90, look at how big that piece of the pie has gotten, right? So a significant improvement there. We see something similar with the other Streptococcus strains, such as Streptococcus rattus. And here, the caries prevalence significantly decreased between baseline and 1 year after the introduction of and use of probiotics containing this strain. Research about probiotics as a health intervention in the gut and oral microbiome, it is promising. Some of it's even very strong. However, I urge everyone in this room as a scientist to use caution because probiotics really are a buzzword, kind of like holistic medicine, natural methods, right? That's being thrown around a lot for use in medicine, dentistry, right? So medical practice, dentistry. Also, again, if we come back to disease prevention and in functional foods. However, information that's provided on marketed probiotic products is not well controlled, which we'll discuss in a little more detail in a minute. But as I described earlier, they're not drugs, right? So a lot of marketed probiotic products can be very confusing. So what do you even look for? What do you do whenever it's not a drug that has clear clinical trial evidence supporting it? And instead, it has a label with claims that generally are not health related, they're structure function related. And you really have to go back to the peer reviewed science to identify whether or not the potential probiotic provides the health benefit that it claims that it will. So, what do I mean by this? Um, it's a species versus strain problem. It really truly is. Remember how I mentioned that Fusobacterium nucleatum has recently been linked to colorectal cancer, but that one of the really interesting findings was that this in the study was that it was only specific strains of the bacterium that actually caused the increase in the tumors, right? They tested 2 different strains, but it was only one that caused the increase in the tumors. The same holds true for probiotics. So if we look here at this study, it indicated that L. reuteri reduces inflammation in the gut. However, if we look at a commercially available strain, so we actually have the strain identifier here, what we see is that the strain didn't inhibit IL-8 production and it did not reduce inflammation. What this means is that a clinician who's interested in using probiotics as an adjunctive intervention or therapy to help support oral or gut health really needs to fully understand the science behind specific strains, not just the commonly reported species in the literature, because there is a major difference. And that issue continues in the market because many probiotic products, this is an actual supplement label, they don't actually have a strain identifier. And what that means is that this strain of Lactobacillus reuteri, which has recently been renamed, but this specific strain, whatever it is, may not be a strain that actually confers that reduction in inflammation. It may be one of the strains that was not shown to have that benefit. This can make it very, very confusing, not just for clinicians, but also for consumers who might flip over the back of the bottle, be very unfamiliar with these bacterial names, and just be like, well, my doctor, my dentist, or whatever said to pick up a probiotic. I just picked up the cheapest one, the quickest one. And that is certainly a concern. What I really want you to take away from this is that research into probiotics as an adjunctive therapy to help rebalance the microbiome is strong. It is growing. There's a lot of interest in this area. Many probiotic strains do produce antimicrobials or biocidins, or they're just much stronger competitors in whatever environment they live in, and they can help support the growth of commensals while reducing levels of pathogens. But strains matter, and microbial interventions require detailed assessments and information at the strain level to assess safety and efficacy. Now, I want to emphasize from a purely scientist perspective, right, which is what I am. I'm a pure scientist. I am on team probiotics. I've seen the science, I've studied the mechanisms, I personally have investigated some of the strains, right? And I truly think that there are bacterial strains that are already commercially available or yet to be discovered that can help support oral health before and after regular dental visits. I'm also a firm believer that probiotics can help patients have a better experience at the chair. But I know that there's a significant amount of misinformation about the health benefits of probiotics, and this includes whether or not probiotics are safe. So again, they are regulated by the FDA. They're regulated as food ingredients, which can gain a status of generally recognized as safe, and I'll explain what that means if you're unfamiliar, or as dietary supplements under new dietary ingredients. The, my issue with this classification is that generally recognized as safe is kind of an open box. Generally, it needs a panel of experts to deem, based on scientific studies, to deem that the product is safe for use in humans, but it doesn't require the level of evidence as something such as a drug would require. Um, Additionally, because of the way that probiotics are regulated, and all you have to show is that they've been either used in human foods and consumption for a long period of time, or that they're safe for human use, what that means is that a lot of companies tend to pick probiotic strains that have a long history of use and not a history of a health benefit. So something like a probiotic, or not a probiotic, I'm sorry, a bacterium used as a starter culture for food products like yogurt is constantly being put into probiotic products for gut health or for oral health simply because it has a history of safe use, not because it has an actual health benefit. So when you get that bottle with the label that says supports gut health in general, what that means is that it's a bacterium that's been shown to be safe in humans and that can survive in the gut, like they use again in something like a yogurt starter culture. And to me, that lack of information is really problematic. Additionally, I mentioned this before, but there's kind of a black box of regulations surrounding probiotics. And so it's unclear exactly what safety should look like. And it certainly differs depending on the manufacturer. So you have to prove in some way, shape, or form that your product is safe if you want this generally recognized as safe or new dietary ingredient status. But the level of what the study, does the study need to be placebo controlled, double blind? Or do 10 people just need to consume something containing the probiotic and not have any negative health effect? It's unclear what that level needs to be. Um, the reason behind that really has to do with this concept of the presumption of safety that we follow in the US for dietary supplements. And what that means, if you're unfamiliar with the concept and you're not sure how it's applied, it means that manufacturers can produce, market, and sell products using ingredients that are presumed safe rather than proven safe. And I really want you to hear the difference between those two words. Presumed safe is enough for probiotic products and dietary supplements. They don't have to be proven safe. And so they're presumed safe until shown otherwise, which would be some kind of a negative health effect. Um, that really comes into play whenever we think about things like antibiotic resistance, right? And I'm quite familiar with this concept based on my doctoral research. Antibiotic resistance is a major healthcare crisis, right? One I'm sure that all of us in here are familiar with. I know that antimicrobial use has really been reduced in the last few years, particularly in veterinary medicine, but there's also been a push in healthcare as well. Um, and that's because commensal bacteria can easily transfer resistance genes to pathogens. It's quite amazing and quite terrifying, especially to see it happen in the lab. Because the US rule is this presumption of safety, antibiotic resistance information isn't even currently required for probiotic strains that are sold in the US, but it is required in other markets such as the EU. And so that's just a consideration that needs to be a part of the decision-making process if you're thinking of using this as an adjunctive therapy. Other safety data that's needed, but doesn't always have to be included, is disease-related data, whole genome sequencing, virulence factors, tolerance limits, right? A lot of people get really excited when they see a probiotic product that markets 5 billion CFUs versus 500 million because they think the more the better. That's certainly not always true. Think about our opportunistic pathogens, generally very unharmful and just perfectly fine in your mouth until there's an overgrowth. So the number certainly does matter. I think that there's a really nice example. So if you need an example of how misinformation on probiotic products can quickly get out of hand and can get confusing for consumers, scientists, clinicians alike. There was this 2011 study where this group assessed commercial probiotic bacterial contents, so what actual strains were inside the probiotic product, and whether or not their labels were accurate. And what they meant by that was like spelling, the organisms that were present, but also the colony forming units, which again, if you or anyone in your life uses probiotic products, they're probably assessed with that CFU number. And I really love this study. Simple. They only did 25 products, but what they found was that the products, not all 25 of them had all the organisms listed. Um, but the two most concerning, misspelled microorganisms on the label. I'm not sure how any consumer should be confident in a product where the organisms aren't even spelled correctly. And then, only 27% of the organisms or of the products had a label claim, had viable organisms that met the label claim. So most of them were quite under. And only 15 of them had a label claim of CFUs, 25 total. So 10 of the bottles didn't even have a CFU claim on it. So you don't even know how much of what you're ingesting on some of these products. Granted, this was done in 2011. I do realize we're 13 years on from that. So things have gotten a little bit better in terms of labeling, but this was a pretty eye-opening study, right? I think for me as a scientist who looks out for these kinds of things and tries to like make sure my family's very aware, but also as somebody who really would like to see probiotic science gain a greater standing in healthcare, but we're held back by issues such as this. And so dentistry, I mean, the folks that I've talked to over the last couple of years since I've been in this position, right, there's been a lot of talk about this huge shift as we transfer, or we're transforming from curative care, treating the disease after it's already occurred and might already be a huge concern, to preventative care. And that there's a really big push to help prevent disease before they even begin. And I think there's a role for the microbiome at the oral and GI level and probiotics for this shift. But it really starts with the patient coming first. And I think that really comes back to implementing things like salivary diagnostics to understand what the microbiome actually looks like, what's actually going on in the oral cavity, and then understanding the science behind probiotics and the different strains to ensure that you're actually helping whatever condition the patient themselves needs. And so I have a list of questions. We recently submitted a paper that goes into a lot of these concepts in a lot more detail, what you should be on the lookout for, the way in which the probiotic market works, and some actual strains that do have clear healthcare benefits based on the research. But we ended it with a list of questions that we think both clinicians and consumers should ask themselves about products if they're considering using them in the clinic. And that includes these four major shortened versions of those questions. One is, does the product label contain the probiotic strains at the strain name level? And does it include an active ingredient dose? Are your CFUs present? And can you actually tell what the probiotic name is? The next is there a history of safety and efficacy easily available for the listed probiotic strain? If you Google the strain, the first things that should come up should not be, I mean, maybe they'll be the company website, right? Depending on how it's characterized and who's paying for what ad at the time. But close to the top, there should be plenty of evidence and studies in the actual literature that show the safety and efficacy and health effect outcomes of these strains. The probiotics, if you're considering using them and applying them, they should provide a health benefit for the specific oral condition of concern and for the specific body area of concern. And then lastly, evaluate whether the product is saying it can claim to treat, prevent, or cure any disease. Because remember, probiotics still are not drugs. If they're claiming to do any of those things, what else are they claiming to do that's inaccurate? Because again, probiotics, they aren't classified as drugs. And so, I just want to, in summary, I just really want to thank you for having me here today, for taking the time to discuss the relationship between the oral GI microbiome and how we can use biological concepts, including things like probiotics, use the microbiome itself for good as an adjunctive therapy to support oral and total body health. And with that, if you have any questions or you want to continue the conversation, feel free to email me. Thank you again to the IDMSS organizers for having me here to speak.